Good afternoon, everyone. Welcome back to the organic dry bean production course. And it looks like almost everyone is in. So thank you for joining us again. This is our fourth installment. I don't know, fourth webinar in the production course series. It's bean time again. Thanks, Rebecca. It's been great having you all participating in the class. And we encourage you again today to ask as many questions as possible. As you know, the organic dry bean production course is brought to you by our Eco Bean project, which is a collaboration between the University of Vermont, Cornell University, the University of Maine, and the University of Wisconsin. And our Eco Bean project is funded through the USDA National Institute of Food and Agriculture through the Organic Research Education Initiative. That's a mouthful. And our team includes researchers and students and extension and farmers all throughout these regions working together to bring you beans. And we've had a great time. We've learned a lot. And we're going to share some of that information with you today. We have one more webinar after today, which is coming up next Friday. So actually a week from today will be the last one. We're skipping Monday. It's a holiday. And so on Friday, we'll be joined by Seth Johnson, a farmer here in Vermont. And he's going to talk about harvest and storage, and plenty of time, hopefully, for Q&A with Seth as well. Again, if you need Certified Crop Advisor credit, Shannon will be putting up a QR code at the end. And if you can't scan it, you can send her your information. And with that, I'm going to turn it over to Ivy for any online updates needed, and also to introduce our speakers today.
All right. Thank you, Heather. And thanks everybody again for joining us for our dry bean course, our class number 4. So if you haven't been able to attend all of them, Shannon has been doing a great job getting all of the recordings up on the course page. So you can go there and watch the past recordings and catch up if you would like to. And so today, like Heather mentioned, we have Dr. Sarah Pethybridge here to talk about dry bean pest and disease management. So Sarah is a associate professor at Cornell University in the School of Integrated Plant Science, and also the principal investigator of the Evade Laboratory at Cornell Agritech. So with that, I will stop sharing my screen so we can queue up Sarah's video. And I'll just encourage folks, if you have questions while the video is playing, put those in the chat. Sarah will be keeping her eye in the chat in case she can answer any questions, and then we'll spend a little bit of time after the presentation answering your questions live before we hear from our research roundup for today, all about dry bean seeding rates.
So the diseases we'll be talking about today fall into 2 groups, depending upon what they're caused by. Firstly, we'll talk about bacterial diseases, but we'll also be talking about the fungal diseases, and specifically the fungal diseases that we'll encounter from here on. And that's probably mostly white mold. So we'll be reviewing some of the symptoms of these diseases, the when and where we might encounter them, and some of the key management tactics that we can do within the season, but also some of the strategies if we've had an outbreak of a disease, what we can do to try and prevent that in the future. So just as a reminder on what is a disease, well, it's not just the presence of a pathogen, our bacteria, or our fungi. We need these 3 factors to occur all at the same time to get disease in our fields. So we need the pathogen, We need the plant, of course, our dry bean plants, and we need susceptible in conducive environmental conditions. And the environment is going to differ. The most conducive environmental conditions are going to differ depending upon the disease that we deal with. And so we'll review some of those conditions more in a moment. So some of the things you may have already experienced with the wet start to the season is the impact upon plant populations or our crop stand through fungal root rots. So fungal root rots manifest themselves as either pre-emergent seedling death, post-emergent seedling disease, so the plant will come up through the soil and then die, and the above ground plant symptoms, if the plants do survive till later in the season, might be stunting of the plants, yellowing of the leaves, generally non-thriftiness of the plant. And when you pull those plants up, you might have these quite dramatic brown necrotic lesions at the soil lines that you can see here. This is caused by a fungal root rot caused by fungus fusarium, whereas this one on the lower side, you can see these brown spots slightly above the soil line as well. These are caused by another soil-borne fungus called Rhizoctonia solani. So fungal root rots, especially in wet, waterlogged soil and maybe wet, cold soil, may be one reason for poor crop stands. They're not seed-borne, they're only soil-borne, and they're generally exacerbated by abiotic stress, such as cold temperatures, and maybe compaction if it's been waterlogged and soil conditions weren't optimal at planting. And generally, soils less than 60 degrees exacerbate root rots caused by these two fungi, mostly these two fungi, Fusarium and Rhizoctonia solani. So by knowing what the pathogens are that cause the disease, we can try and design crop rotations to try and minimize their effect on future dry bean crops because they're soil-borne only and not seed-borne. But one of these fungi, Rhizoctonia, also may cause issues later in the season. So if you've had issues with poor crop stands due to Rhizoctonia, there's probably a higher risk of seeing foliar blight. As you can see here, these sort of very dramatic bleached type lesions scattered throughout the foliage, also caused by this fungus. And if the pods are more likely to contact the ground, either through a variety or crop lodging or through a storm, for example, has pushed the canopy over, you may also see disease caused by Rhizoctonia on the pods, as you can see here. So sort of bleached looking lesions, random spots across the pods. So again, that's an indication of Rhizoctonia present in the soil. And if that's an issue, we can certainly take that into account in our crop rotations in the future as well. So in terms of root rot management, there's not a lot that we can do from now on. Our plant populations are already set. So there's minimal within season intervention, but it's really strategies, things that we can do prior to planting, which are going to try and reduce our crop loss caused by root rots in the future. So if it has been problematic in certain fields in the past, we can try and incorporate that information into our crop rotation plans. Other diseases that you may have seen already in your dry bean crops and may continue to see throughout the season are those caused by bacteria. Now, there's 3 different diseases caused by bacteria which are more common in the northeast. The first of these is brown spot, and it's caused by a bacterium known as Pseudomonas syringae. It causes these interesting lesions on the leaves. They're brown or necrotic, what we call, and you can tell that they may be caused by a bacterium because of these yellow rings or yellow halos around these symptoms. So you might see symptoms of brown spot on the foliage, but you may also see them on the pods, as you can see here. The lesions don't get very large. They generally less than around 1/8 of an inch in diameter. They're generally circular, but they always have this yellow ring. They can sometimes often occur in lines or veins along the leaf because the leaf wetness will accumulate in those areas and therefore exacerbate infection. And when those lesions dry out, you can get the center of those lesions falling out, and the leaf might look like has a shot hole type appearance, as you can see one here that's more advanced and cracking compared to the others. Now, there's two other bacterial diseases you may also encounter. The first of these is halo blight. It's a lot more dramatic in its appearance rather than just small necrotic lesions. It's caused by a similar bacterium, again, another Pseudomonas syringae, but a different type of bacterium. And the lesions are generally a lot larger, the yellow to green halos around the lesions again, which is characteristic of most bacterial diseases, but they're a lot larger. Their upper side is a red type color, and again, they occur on the foliage and the pods. Now, you may also encounter common blight, which is caused by a different bacterium. This one's a Xanthomonas. And again, the symptoms occur on the foliage and the pods. They usually start off as small water-soaked lesions on the undersides of the leaves and then progress to those brown, dry and brittle type symptoms that were similar to those other bacterial diseases. But it may cover a larger area of the leaves compared to brown spot, which are usually quite small lesions. This one is a lot more damaging than the other two bacterial diseases. It can dramatically impact yield, reduce the number of pods, and of course, we may also encounter poor seed quality within the infected pods as well. So in general, these bacterial diseases are necrotic lesions with that yellow halo, and differentiating between them can be quite tricky, but it's generally the size and appearance of the lesions. Now, for all of these bacterial diseases, the pathogens generally survive in crop residues, so residues of previous crops which may also be hosts. They can also be seed transmitted. They can be soil-borne, and they can survive on weeds as well as alternative hosts. But in terms of crop rotation, the key information is that the host range mostly consists of all beans. They don't discriminate against beans. So it's lima beans, snap beans, soya beans, which is probably more important in our rotations, and broad beans. So making sure we've got a rotation away from other beans, not just dry beans, is also important for planning crop rotations in the future. The spread among and between fields, so the dispersal of the pathogen, is usually driven by wet weather, hail, rain, windstorms, everything that we've mostly been encountering this season. So perfect weather for bacterial disease spread this year. But the weather will also drive the conducive conditions for the disease. So for brown spot and common blight, they tend to be exacerbated by warm temperatures, so 80 to 85, whereas halo blight is favoured by cooler temperatures, 70 to 75. So really there's a bacterial disease out there for every different situation. I would if I was a bacterium, I would certainly like the 70 to 75 rather than the 80 to 85, but they seem to cover all bases. So in terms of bacterial disease management, things that we can do within the season, if we do have a problem or watching the problem, the most impactful thing really is to get on it quickly. So if we do have an outbreak of bacterial diseases, knowing that it's out there. So scouting regularly is really important because these diseases can spread quite rapidly, and especially in the environmental conditions we're experiencing this season. So really, if we want to try and prevent the spread of bacterial diseases, no matter what bacterial disease we're dealing with, the strategy is almost the same. So generally our only option for sprays are copper-based sprays, and they're generally put on mid-vegetative or early flowering periods, so getting to the stage now. Because they're copper-based protectant sprays, one of the most important things to getting the most out of your investment in copper is ensuring good spray coverage. So if you've got a thick canopy, making sure you've got the right water volume and the most appropriate nozzles for getting spray penetration into the canopy. Now, of course, there's coppers for conventional production and there's coppers for organic production. So those are formulations that are OMRI listed. And we've done a lot of trials over the years with different coppers and different diseases. And we believe that generally all coppers are the same as long as you use them at the maximum labeled rate in terms of their disease control efficacy. Now, there are differences in terms of maximum labeled rate, cost, mixing, usage, the ease of usage in mixing. So some may block nozzles compared to others. But as long as you use them at the maximum labeled rate and treat them as protectants, they're not going to cure the disease. They're only going to protect future infections. You will get the most out of your investment. The other disease you might encounter from mid-season now to harvest is a disease called anthracnose. Anthracnose is a fungal disease. It's caused by a fungus called Coleototricum lindomathium. And these are the typical symptoms that you might see on the foliage. So they're rust brown to black lesions, mostly on the petioles of the leaves and possibly the leaf veins. And they're more round but sunken eye spot type lesions. They can be particularly aggressive and spread quite rapidly. So if it's earlier on in the season, it can also kill individual plants, but usually they cause general defoliation and unthriftiness. But anthracnose can also get into the pods, and it can cause these quite dramatic black or brown type lesions. And those black and brown type lesions are usually associated with these orangey colour around them. And those are the spores of the fungus, which are going to be dispersed to infect other plant parts. So overall, the major impact of anthracnose is on pod quality and seed quality as well. So one of the characteristics of anthracnose is, again, similar to bacterial diseases. The spores are spread by rain splash. We're encountering frequent moderate rainfall, which is conducive to the disease. It's got a very wide temperature range, so it's happy either way, right down to 55 degrees and up to 80, but it really likes high relative humidity, over 92%. Now, things that we can do within the season. Well, we could apply fungicides if it's becoming an issue. And a lot of the fungicides that have efficacy for white mold, which I'll talk about more in a moment, are also going to pick up control of anthracnose. So I'll talk more about optimal timing for fungicides in a moment. And then planning for the future, thinking about crop rotations as well, away from beans for at least 3 years, all beans, not just dry beans. But one of the most damaging diseases affecting dry beans from mid-season to harvest is white mold, of course. White mold is caused by the fungus Sclerotinia sclerodiorum, and one of the major reasons it's such a problem is that it has a very broad host range. So crop rotation is not always effective, as it could be in other systems, to try and break that inoculum carryover cycle between seasons. So it infects many of our field crops, vegetable crops, and our broadleaf weeds as well. So I'm sure if you're a dry bean grower, you would have been familiar with white mold symptoms, but here's a picture of some. There's bleached brown lesions, as you can see here, on the stems and the pods. It doesn't usually affect the leaves directly. And the telltale thing that it's white mold is that we're getting the production of this white cottony mycelia on those diseased plant parts. And after you get the production of this white cottony mycelia, you get the production of the black sclerotia. Now, the sclerotia are the resting bodies of the fungus that fall back into the soil surface, and in that way it can survive between seasons to cause disease in subsequent crops. So generally on the stems and pods, but sometimes when you're scouting, it's hard to see those symptoms because they're way down in the canopy. But sometimes you have a flag leaf, so a leaf that's sort of wilting singly on a stem. That indicates that there's generally something wrong lower down in the canopy. And often when you part the canopy, then you can see white mold symptoms. So sometimes the most obvious things to look for are those flag leaves at the top of the canopy. Now, white mold, again, bringing in that disease triangle concept, pathogen, Sclerotinia sclerotiorum, is usually present enough in the soil because of that broad host range to cause disease if the conditions are conducive. And in dry beans, there's not a lot of significant resistance or immunity to our cultivars, which would change our crop protection practices. So it's really the environment that drives whether we get white mold in a particular year or not. But often it's this white mold mystery, from what I say. It's when to get the most out of your crop protection practices. Often there's a mystery about where it's going to occur, trying to predict whether you're going to get white mold or not. And then the next part of the mystery is how. What are we going to select as the most effective crop protection product if we decide to use those for white mold management throughout the season? So let's explore the when, where, and how in further detail. So often it's a decision around whether to spray a fungicide or not. And to get the most out of those fungicides you're investing in crop protection is to use them as protectants and not curatives. So often if you're already at the stage of seeing white mold, you've already had a lot of infection, those fungicides are not going to cure the infection, they're only going to protect the further developing flowers. And I'll get to that more in a moment. So this is the life cycle that is interesting to understand to get the most out of your fungicides. So those sclerotia, the black resting bodies of the fungus in the soil, germinate. They then produce spores, which fly off and are wind dispersed. But the only way they can infect a bean plant is through the flowers. It doesn't matter if there's lots of foliage. If there's no flowers, they're not going to infect, because they infect the flowers, use those petals as when they're dying as a nutrient source to infect the foliage. So we can use this information to try and get the most out of our fungicides. We need to protect the flowers. So when judging the long-term risk of white mold, whether you're going to get white mold in a particular field or not, we can take into account the history. So if we've had susceptible crops in that field, have they had white mold before? If they have, of course, then there's a higher risk because there'll be more sclerotia present in the soil. Now, also topography also influences white mold risk. So the undulating landscape that we experience in New York and Vermont and elsewhere is perfect for enhancing the conditions for white mold. Poor air drainage around sites that might be surrounded by trees, for example, anything where relative humidity is high and leaf wetness is going to extend, also make the field more conducive for white mold, assuming the inoculum is present in the soil. So when drilling that down further, The risk factors for white mold are, well, it likes cool conditions like me, less than 85 degrees Fahrenheit, a normal or above normal rainfall, similar to this year, soil moisture at or above field capacity. But if it's too wet, that can also be deleterious to the survival of the sclerotia. So if you have flooding, for example, that will kill the sclerotia because they like oxygenated environment. Morning fog and high relative humidity, which keeps the leaves wet, are also exacerbating white mold. So in terms of the risk factors to the crop, a cropping history, like a higher frequency of crops in that field that will be susceptible to white mold, which is most of our field and vegetable crops. If the particular crop has an early or dense canopy closure, high plant populations or excessive plant nitrogen, that also exacerbates the risk of white mold. And weed management, of course. So if there's a lot of broadleaf weeds, even when we've grown non-susceptible hosts, like corn and cereals, then they may also serve as alternative hosts to keep the inoculum going in that field. But overall, cool, wet weather is favourable for both the pathogen and the crop because it often extends the flowering window in dry bean. And even though dry beans tend to already flower for quite a long time, that could be extended and there could be more flowers because flowers, again, are the only infection point for the fungus. So often there's a lot of debate about, well, where is it going to occur? There has been some apps developed from the Midwest, and Sporecaster is one that was developed at the University of Wisconsin. So what Sporecaster does is evaluates if the weather has been favourable for the germination of the sclerotia into those mushroom structures which produce the spores. So it takes into account a 30-day average of the maximum temperature, relative humidity and maximum wind speed for drying the canopy and dispersing the spores. And all you have to do is provide in the app the GPS coordinates of your field, and I'll show you more about that in a moment. This app was developed for soybeans, so the crop parameters that you can alter are more related to soybean production. So you can say whether it's irrigated or not, and whether it's on a 15-inch or a 30-inch row spacing. So not as ideal for dry beans, but it's an indication of whether you're going to have weather that is conducive for sclerotia. So just as a reminder, fungicides are protective, they can't be curative. And if investing in crop protection, trying to protect those flowers from infection by sclerotinia is the key to getting the most out of your fungicides. Now, if you're in organic production, we've also done a lot of screening looking at the efficacy of different OMRI listed products for use within the season. And the principle is the same, to try and protect those flowers from infection. Now, the ones we've had most success with contain a Bacillus amylocofaciens strain. And here's two different strains with two different formulations from two different companies. One's called Double Nickel from Certis Biologicals, and Stargus is from MBI. We didn't find any difference between them, but we found that these particular active ingredients were the most efficacious for the control of white mold. We tend to avoid copper over flowering in dry beans because it can be phytotoxic and cause premature flower abscission with high temperatures. So that's why we were looking at a lot of the different microbial biopesticides for white mold control over flowering, whereas copper on the foliage will be your best option for bacterial disease control, of course. Now, those are things that we can do within the season. Of course, we're also looking ahead and trying to be proactive to try and reduce the risk of diseases in future years. So looking ahead to 2025. Some of the most impactful things are using certified disease-free and treated, if possible, seed for conventional production, obviously certified seed for organic production. So making sure that we're not introducing some of those really aggressive seed-borne pathogens into our field to start with, so we don't even have to deal with them. Crop rotation is another completely impactful production practice for a lot of these pathogens. So a lot of these pathogens have beans in general as their host range, as we saw with the bacterial pathogens, anthracnose. This is not the case for white mold, but for some of those other pathogens, rotating away from beans for at least 3 years is our recommendation. Now, if we've had an outbreak of white mold this year, we can look at ways to try and minimize the impact or minimize the carryover of that inoculum so it doesn't cause as much disease as it could in subsequent crops in future years. So for sclerotia, the resting body of the fungus that causes white mold, we recommend not leaving them on the soil surface. And if you can, do some shallow tillage to try and cover them with soil, that promotes their degradation. And in our trials in New York, that promoted degradation in a faster rate than if we left them on the soil surface subject to the elements. And of course, crop residues management also comes into our thoughts about what we do after a disease outbreak. So many of those pathogens, anthracnose, the rhizoctonias, the bacterial pathogens also survive in crop residue. So we also encourage you to consider incorporating them promptly to try and encourage decomposition at a faster rate, similar to the strategy for sclerotia.
Awesome, thank you. I'll thank Sarah again for putting together that presentation and sharing some information on dry bean diseases. If Sarah was going through the chat while the presentation was happening and was answering some of the questions that folks had, if there's more questions, feel free to put those in the chat. We have time for a couple before we jump into our research roundup. Sarah, I don't know if you have anything you want to share or add on in addition to what you shared in your presentation, while we wait for some questions.
No, they were really interesting questions in the chat. Thank you.
Someone in one of the earlier webinars had asked about seed treatments. We were talking about inoculating beans at planting, but then they were wondering if there was seed treatments to consider, because you talked about, planting clean seed is really important. So we saved that question for you.
Right, yeah. We've been testing a range of different biological type products that could be used as a seed treatment, mostly targeting anthracnose, and we haven't found any that have been effective, unfortunately. We also tested an OMRI-listed copper, and they were also phytotoxic to seedling growth. But Heather, have you been doing any similar work?
Yeah, I mean, we have been looking at treatment of seed-borne diseases more specifically, and we've been trying to use ozone treatment as a means to reduce seed-borne diseases. And then we've also tried or working on trying, other techniques as well, but we don't have any recommendations at this very moment, but I think it's on our minds. We know it's an issue. And I think Kelly's on, who's been working with us on the ozone treatment. And we got that idea from one of the farmers that was growing beans.
Okay.
Because he was having so many issues. So it was a strategy that he was trying to use. So the verdict is out, I guess.
Yeah.
Yeah. I do think people testing their seed lots is really important, and one of the things that we've been trying to help people with, and some of you on here may have submitted seed to some projects we've had to look at seed-borne disease in dry beans, and kind of trying to figure out what the extent of infection is and severity. And I think it's been brought up about accessing seed, especially heirlooms, specialty types, and people saving their own seed, and just making sure that you're monitoring diseases in that seed that you save yourself. So I don't know how helpful that is, but we have been helping people with seed-borne disease testing, at least so they know,
Mm.
what's going on with their own seed lots.
There's a lot of interest about Mexican bean beetles.
I know.
Ballistic beetle.
Yeah. I've gotten so many questions on that this year. And we don't have a ton of answers, but I was definitely thinking, Sarah, and if we continue this work, that's a topic we really need to dive into. Seems to be getting worse.
Yeah, in New York, we've also had similar experiences with specifically Mexican bean beetle as well. Yeah.
Yeah, that was.
Oh, go ahead, Ivy.
No, there was a question. I might have been the same one coming back around. Just like plant nutrition. I think in Kristen's presentation, she pointed at talked about the concern of excess nitrogen, maybe exacerbating disease in your dry bean crop. I don't know if Sarah, you have any more information about that.
Yeah, I mean, probably, if you've got excess nitrogen, you're going to have more prolific canopies, which may make the conditions within the canopy, more conducive to infection and disease development. So I guess it's all a balance. For many of these diseases, we're trying to promote airflow through the canopy to reduce infection. So, optimizing that, especially the nitrogen levels, will be important.
Mm-hmm.
Yeah.
Also, either I could put it in the chat or Shannon might be able to. Someone asked about resources for monitoring saved seed. I mean, we have Kelly and our team put together a guide for farmers with that project Heather was talking about. So we can put that in the chat as well. But I don't know if Sarah, if you have any other.
No, I think
insight.
no. I mean, you guys are a great resource for that.
We can also.
Sorry, Lynn's question about blind flaming. What do you mean by blind flaming, Lynn?
I wonder, are you referring to flame weeding, like using propane weeder? And you can unmute if you want, Lynn, and ask. Anyway, yeah. I wonder if some of the thought behind that is, if you're using a flame weeder, if the heat itself would do any damage to sclerotia possibly, or there we go.
Heather, I finally figured out how to get to my machinery to work. What I'm talking about is something used more typically in the Midwest, where we use a propane-based flamer
Yeah.
to treat all of the ground, even above the rows, just before emergence. So that's called blind flaming. There's a group of heat transfer experts at the University of Nebraska who have fine-tuned flamers and know how many milliseconds any weed or a sprout needs to be killed. I was wondering if that helped at all in minimizing spores and minimizing fungal diseases.
Yeah, I mean, probably the major impact of that might be if you're contacting crop residue that might be harboring the fungal spores or the mycelium that may be carrying over that inoculum between seasons. And we've done a few experiments where your equivalent of blind flaming is after harvest. So we're trying to break down the residue for those fungal species that only infect the residue but don't survive in the soil on its own. So I would expect if there's similar strategies, like if you're breaking down the residue faster, firstly, you could have that indirect effect because there is no substrate for the fungus to survive on anymore. But there may also be that, if you contact those propagules of the fungus directly, if you get high enough temperatures, then you may have a direct toxic effect as well. I'm not sure about sclerotia. They're a really tricky one. They survive anything. And also when you do have tillage, a lot of the sclerotia from further down in the soil profile will be brought up again. So those are unlikely to be contacted by flaming on the soil surface. So when you do provide tillage, you're just going to get another crop of sclerotia.
Right.
Well, one of the points of flaming is to avoid tillage. So the
Oh,
last tillage pass is really when you're planting.
right.
So you're not using rotary hose, you're not using cultivators, you're just using flame.
Yep. Yep. Right.
Thank you for your presentation. I've enjoyed it.
Thank you, Lynn.
Just reading one of the questions, if anyone's used pre-plant field flooding as a tool for reducing in-soil pathogens.
Yeah, so sclerotia of Sclerotinia scleratiorum will not survive in flooding conditions, because they need oxygen in the soil to survive. So the anoxic conditions of flooding does promote reductions in sclerotial viability, but I'm going to defer to Heather for the effects on soil health.
Yeah. Well, it depends, I guess, as far as soil health goes, what kind of sediment you're getting from the flooding. Certainly other issues with, reducing biological activity, at least in the short term and things like that. But I'm just looking at the question. Yeah, I I had never thought about that, flooding something on purpose. Which is a good point. I mean, it happens other places where people are using flood irrigation and things like that. So that's a really good question.
Yeah, in answer to your question, Patrick, about how long does a field need to stay flooded? They've used this in lettuce crops in Australia where it's quite sandy soil, but they found they had to keep the soil flooded for 4 to 5 days.
Interesting.
So it's quite a significant period.
Yeah.
In answer to the question about running the crop residue through ruminants, unfortunately, sclerotia will survive through a ruminant as well. So that's again the resting body of the fungus.
Thank you. I think so. It's about 12:45. So if folks still have questions, that's great. You can put those in the chat, but I think I'll introduce our research roundup speaker so we can hear from him. And then after that presentation, we'll still have time for more questions. So Adam, if you want to get your slides up, I'll introduce you. So for our research roundup today, we have Adam Sharifi, who's a master's student in the Sustainable Cropping Systems Lab at Cornell University. And so Adam's going to talk about research that he did on seeding rates in organic no-till production. And so this was during his undergraduate years at Cornell. And so we're going to hear about his research now.
Awesome. Can you guys see my screen all right over here?
Yep, that looks good.
All righty. Okay, well, good afternoon, everyone. My name is Nazir Adam Sharifi, graduate student here at Cornell Sustainable Cropping Systems Lab. Pleasure today to present some of my undergraduate research on effects of seeding rates in organic no-till black bean production. All right, so in the Sustainable Cropping Systems Lab, we do a lot of work in cover crop-based organic no-till systems. And for anyone who isn't familiar with this approach, let me briefly walk you through how this works in the context of our black bean study. So in the start of the fall, we start by planting a cover crop, in this case cereal rye, which grows and overwinters until it reaches a suitable growth stage for termination in late May. And then at this point, we terminate it by using a roller crimper, which is a steel drum with blunt blades mounted on front of a tractor that kills the cereal rye by crimping it and flattening its stems, so it leaves behind a thick mulch on the soil surface. Simultaneously, we're planting black beans directly into the mulch using a no-till planter that cuts through that residue and places the seed in the soil. And after a few months, the goal is to have a strong, uniform black bean stand like you see here. And while the beans are definitely the star of the show here, the cereal rye is doing a lot of that behind the scenes weed suppression. The mulch suppresses weeds in several different ways. It moderates soil temperature and moisture, can release allelopathic compounds, can also tie up nutrients such as nitrogen, making them not accessible to weeds. And the most obvious here, physically blocking out light and making weed emergence more difficult. But even with all these benefits, weeds can still break through the mulch and persist. And since tillage isn't part of this system, additional weed management strategies are needed to improve its overall weed suppression. So this is where cultural weed management practices come in as a way to strengthen weed suppression in our organic no-till systems. Examples of cultural practices can include things like seeding timing, like changing planting date, narrowing row spacing to speed up canopy closure, and we'll focus on today, which is increasing seeding rates. So this idea that higher seeding rates improve crop competitiveness at a population level. So quite literally, it is the idea of power in numbers. There has been previous research that shows that higher seeding rates can improve weed suppression in several crops, including corn, rice, and wheat. Our lab has also seen this effect in organic no-till soybean systems, where increased seeding rates can enhance weed suppression, yield, and profitability. So this approach has definitely shown promise in organic no-till soybean, but has yet to be tested in dry bean. And in our earlier work, we found that dry bean yields in organic no-till systems were lower than in our tilled systems. But this was when both systems were using the same seeding rate. So this led us to ask whether higher seeding rates in organic no-till dry bean could improve agronomic outcomes compared to the current recommendations. So the objective of this study was to optimize production of organic black bean no-till planted into rolled crimped cereal rye. We hypothesized that increasing black bean seeding rate would increase crop density and improve weed suppression by reducing weed biomass. We also expected higher seeding rates to increase yield and profitability. At the same time, though, we expected some trade-offs at these higher populations, specifically greater risk of white mold and increased lodging. So this experiment was conducted across 5 locations in the Northeast and Upper Midwest. So sites include Alburg, Vermont, Arlington, Wisconsin, Aurora and Geneva, New York, and Stillwater, Maine. The study was ran in 2022 and 2023, with the site in Maine only conducted in 2023. So this gave us a total of 9 site years of data. And for our experimental design year, we used a randomized complete block design with 4 replicates. We had minimum plot sizes of 10 by 20 feet with 4 rows spaced 30 inches apart, so 30-inch row spacing here. In this table, you can see the seeding rates we tested ranging from 60 to 375,000 seeds per acre, with a 0 seed per acre subplot in our lowest seeding rate treatment to really examine weed growth without black bean competition. All right, let's dive into some data here. This is the format that we'll be using for the results. 9 panels, each panel representing a single site year. And we decided to show the site years separately because we believe each one of them tells its own story, as you'll see here in a moment. So on the x-axis here, we have black bean seeding rate. On the y-axis, we have black bean density measured at harvest. The dotted line in each panel represents the expected density based on 89% germination for Zorro black bean based on the seed tag. And across site years, as seeding rate increased, black bean density also increased, though the degree of increase varied. For example, Aalborg 2022 and Geneva 2022 had very high stand establishment, while Geneva 2023 and Stillwater 2023 had definitely lower establishment. We attributed reduced stands across these site years to a few factors. For one, planting through a thick cereal rye mulch can definitely reduce seed to soil contact on occasion, leading to limited germination. There's also lower soil temperatures under the mulch, which can also delay emergence. And another thing that we were looking at was soil moisture at planting that varied across sites. So some site years were very, very dry around planting, and others were very wet and cold. So we believe that these factors together definitely influence the stand establishment that we're seeing here. Now let's look at the effects of black bean density on weed biomass, with black bean density on the x-axis and weed biomass on the y-axis. So across our 9 site years, we saw a clear reduction in weed biomass, higher densities in 4 of our site years, and those are the ones that are not shaded out here. Interestingly, these were the site years with moderate to high baseline weed abundance. And when I say weed abundance here, I'm referring to the biomass measured in those 0 seed per acre plots. Essentially, the weed biomass in absence of crop competition, or really how weedy this site really was. So to be specific, site years with a baseline weed biomass greater than 1,100 pounds per acre showed reductions in weed biomass as black bean density increased. And this will be a reoccurring theme throughout this presentation, especially when we're looking at yield and profitability. Next, let's look at the effects of black bean density on white mold incidence. So black bean density is on the x-axis here. White mold is on the y-axis. And we found that higher black bean densities increased white mold incidence in 2 of the 9 site years, which were both in Geneva. And this kind of harps on a little bit of what Sarah was talking about previously, but this increased incidence of white mold in Geneva can be traced back to its field history. So this site was inoculated with sclerotium in 2019 for a previous study, and this pathogen can really persist in the soil for a long time, maybe sometimes up to 8 years. So these results suggest that in fields with a known history of white mold, higher seeding rates may increase that disease risk. If we look closer here at Geneva 2022, we can see that the current conventional practice, white mold incidence, was about at 13%. But when we roughly double that density, that incidence jumps to 50%. So again, denser plantings create a more favorable environment for the pathogen, as these denser canopies traps more moisture and humidity that favors this disease growth. And we also looked at lodging, rating it from a scale from 1 to 5, where 1 was no lodging and 5 was complete lodging. And while I don't have a nice fancy figure here to show this, we did see that increased black bean density did increase lodging severity in 3 of the 9 site years. So together, these results kind of highlight some of the trade-offs maybe of increasing seeding rates with these greater risks of white mold and lodging. Now let's look at the effect of black bean density on yield. Black bean density is on the x-axis and yield is on the y-axis. That yield is adjusted to 14% moisture. And across the 9 site years, increasing density increased yield asymptotically at 5 of our site years. In the other site years, yields plateaued quickly at the lower densities, so increasing density didn't really result in any significant yield gains. Similar to what we saw with weed biomass, significant yield gains occurred in site years with these moderate to high baseline weed abundance. So in this case, where baseline weed abundance was above roughly 1,000 pounds per acre of weed biomass. And to illustrate this a little bit better, let's take a closer look at Aurora 2023. So in this figure, the black line represents average weed biomass at the site. Colored lines show simulated scenarios. So the red line is high weed abundance, and this is 150% the average weed biomass of that site, while the green line shows low weed abundance, so 50% the average weed biomass at that site. And at low weed abundance, that green line, we see that the yield plateau happens quickly at these lower densities, where at high weed abundance, that red line, yield increases more gradually and requires these higher densities to reach maximum yields. And so this kind of shows that these higher black bean densities are particularly beneficial when weed abundance is high. And I just want to mention another quick interesting pattern that we saw, and that is that the maximum yield achieved at that yield plateau varied across our site years. And we found a strong linear relationship between the height of that yield plateau and the total growing season precipitation. So in the graph to your right, each point represents a site year with precipitation on the x-axis and maximum yield on the y-axis. And the takeaway here is that that yield plateau at high black bean densities was greater in site years with more growing season precipitation. All right, now let's look at the effect of black bean seeding rate on partial returns. So seeding rate is on the x-axis here, and partial returns, which takes into account seed costs, are on the y-axis. And we used a quadratic model here to describe the relationship. So the dotted line, the dotted vertical line, shows the conventional recommended seeding rate, and the solid line that's vertical there shows the economic optimal seeding rate for each site year. And this model shows that as you increase seeding rate, partial returns increase up to a certain point, and then beyond that peak, returns start to decline because these additional yield gains no longer offset extra cost of the seed that you're buying. However, this quadratic model didn't really fit all site years equally well. So we used some kind of stats jargon here, but we used conditional R squared as an indicator of model fit, so how well that quadratic fit. And site years with a R squared value below 0.30 were considered poorly fit by the model and really not suitable for interpretation. But in the remaining site years that were adequately described by that quadratic model, optimal seeding rates were equal to or greater than 230,000 seeds per acre. However, there was this exception here with Arlington 2023. Even though the model fit well, the optimal rate was below 230,000 seeds per acre. So this kind of raises this question of, okay, why are some site years having these bad model fits, and why is Arlington 2023 have this good model fit, but this lower optimal seeding rate? And we determined that these higher optimal seeding rates at 230,000 seeds per acre or greater were only justified in site years with adequate stand establishment and moderate to high baseline weed abundance, roughly 1,000 pounds of weed biomass per acre. And these conditions lined up with site years that didn't meet our criteria. For example, Geneva 2022 and Stillwater 2023 had both poor model fit with profitability model, but they were also the site years that had the weakest black bean establishment. And then when looking at site years like Arlington and Alberg 2023, they had a very low weed abundance, which also explains why they didn't have these optimal seeding rates greater than 230,000 seeds per acre. So for site years that met the criteria for high seeding rates, economic gains relative to the conventional rate ranged from $261 to $400 per acre. So some yield gains there from the optimal seeding rate. And this really shows some of the potential profitability aspects of increasing your seeding rates under the right conditions. So in summary here, for organic no-till black bean production, we found that higher black bean densities reduced weed biomass in site years with moderate to high weed abundance. that white mold increased at higher densities in Geneva due to field history of disease. That lodging also increased at higher densities, again, highlighting some of these trade-offs with higher seeding rates. And yield benefits of higher black bean densities increased with wheat abundance, as we saw in that multicolored figure. And then finally here that seeding rates equal to or greater than 230,000 seeds per acre really maximize economic returns under these weedy conditions. So if there's one thing that we take away from this presentation today, I would say that it's for organic no-till black bean production, that increasing seeding rates beyond conventional practice can improve yield and profitability, but benefits definitely depend on weed abundance and require these site-specific adjustments to manage white mold and lodging risks. So yeah, thank you for your time, and here are some of my acknowledgments. Happy to take questions if we have time here.
Thank you for sharing all that research with everybody. And we do still have time for questions. We have about 10-ish minutes before we have to wrap up. So if folks have more questions for Sarah or for Adam, please feel free to put those in the chat. I don't know, Adam, if you want to speculate. Patrick had a question about if the high seeding rates in pinto beans would result in similar results.
Yeah, and apologies, it looks like I'm a little bit under the weather here, turned yellow, but I would be hesitant to speculate. I feel like each variety can be different, and also depends on growth habits as well. We use kind of a type 2 architecture here for our plants, and I'm not too well versed, I guess, with pinto bean, but if others would like to comment on that, please.
And then for the test sites for those seeding rate trials, someone, I think they're from like the Western US, were they all at relatively high humidity sites compared to like the Western US?
I would say so. I feel like in the Northeast we definitely experience these higher humidity levels, and we also receive a lot of rainfall around these parts too. So I would say like for our New York sites I can speak to because those are the ones that I was frequenting more often. We definitely receive a lot of rainfall, and paired with that humidity, it's definitely a beautiful combo for white mold. So yeah.
Mm-hmm. Yeah, I know our site in 2023 was exceptionally wet. So yeah, you see that that moisture getting trapped in those higher seeding rates for sure. And I'll just keep looking through the chat here. So I apologize if I missed any questions from earlier. Just trying to see.
A question about is weed biomass on a dry matter basis or wet weight, and this was dry matter. So yeah, thanks Ivy for commenting on that. I appreciate it.
Yep. I know earlier on in the presentation, Rebecca had asked Sarah, while Sarah was presenting, they were curious about, Mexican bean beetle and some other pests. One of the ones Rebecca asked about was seed corn maggots. I don't know if Heather, if you have any input on how someone could deal with that, or if it's of a major concern.
Yeah, I mean, seed corn maggots can be a major pest in beans and dry beans, as well as soybeans, lima beans. All of those, they seem to have a really strong affinity to lima beans. And the seed corn maggot itself it's an optimist, an opportunist. Maybe it's an optimist, I don't know. But it's after like decomposing plant material, really. And so if beans are planted into, I would say, good conditions, which we heard already from Kristen, we're talking 60, 65-degree soil, adequate moisture, and warming conditions, and the plants themselves are able to germinate quickly and start growing. Generally, seed corn maggot should not be a significant issue. But if you're planting into cool, wet, damp soils, that the potential for prolonged time to emergence, then those plants certainly could get eaten up or damaged by seed corn maggot. So really the best way we have to combat that pest, especially organically, is by managing planting timing and conditions, which isn't always that easy. And then outside of that, we also know that high amounts of decomposing organic matter residue followed by planting can also lead to more damage. So if you do have a lot of plow down cover crop and you're using tillage or some kind of compost or manure, that can lead to higher levels. So just waiting for a time period could help reduce that pressure.
Thank you. I see Kelsey had a question. I think it's for Adam. And I think I understand. Do you expect that we would see similar results with another crop other than rye? Maybe a cover crop that winter kills.
Yeah, that's an interesting question. If I think about it this way, so I would say no, only because so cereal rye is this very unique in the sense that it produces a lot above ground biomass here. And depending on what winter kill cover crop we're talking about, it might not be that same amount of biomass. So if we're talking about weed suppression of the overall system, it's definitely going to be higher in our cereal rye system, which would maybe justify for seeding rates on, I guess, the lower end compared to maybe a winter killed system where not a lot of above ground biomass. So you're going to need potentially maybe even a higher seeding rate to optimize your weed suppression there. So I'd say definitely maybe some differences. I would also like to comment that we did have this experiment done in a tilled system as well, so an organic tilled system. Seeding rates there were different. We actually saw that in our tilled systems, there was about, I think, 200,000 seeds per acre seemed to be around the optimal seeding rate, and that was with field cultivation as well. So definitely, unfortunately not like a nice clean blanket seeding rate for all conditions, but as is agriculture, I guess, right?
And Adam, I think, did you write up a report summarizing like the multiple sites with the tilled and the no-till seeding
Yes,
rate
I did.
trials?
Yes.
We can definitely share that with folks, just as a good reference to read. There's a lot of good information in there, so put a plug in for that, and we'll put it on the course site as well. So we have about 5 minutes left. Shannon, I don't know if you can share our slides. We'll as we wrap up here, if folks do have any more last minute questions, feel free. You can put those in the chat while we end our session here. So like we had mentioned earlier, we have CCA credits available. So if you need those, feel free to scan this QR code here, but Shannon can also help you out if you need help getting those credits. And then, like Heather mentioned, today, this Friday is our 4th class. We have one more class coming up next Friday. We're going to skip Monday because of the holiday. So if you go to the next slide, Shannon. Yep, so On Friday, February 20th, we're going to talk all about harvest and storage of dry beans with Seth Johnson, who's a farmer here in Vermont. He has a lot of information and experience growing, harvesting, cleaning dry beans. So it'll be a really good presentation to hear from him. And then our research roundup, we're going to dive back into dry bean diseases. So we're going to hear about root rot pathogens in dry beans. So there's a lot of good information still to come. And we do have a short survey for folks. If you could just fill that out after we wrap up here today, that would be really helpful. I did see a couple questions just pop up now. Let's see if Adam did a parallel study with the soil, the sites you were doing. Not
Yeah,
quite.
I can quickly answer that. I
Okay.
think if the question is, if I'm interpreting correct, we did replicate the study, but only at the Vermont and Aurora sites. So didn't do it at all. Like there wasn't 9 site years of data. There was 4. So hopefully I'm answering your question correctly there.
Thank you. And then I don't know if you can answer this. Mark asked about ideal seeding rates in 15-inch rows. They currently plant 225,000 plants per acre.
Yeah, that's a good question. I will be transparent here and say that I've only worked in 30-inch rows. So yeah, if I had some time to logically think through that, maybe I could come up with an answer here. But yeah, based on my experience, I don't think I can provide credible feedback here.
Thank you. And I'll thank Sarah and Adam again for taking the time to speak at our class today and share all of their information. And the presentations will be available on the course page as well as the slides. So those will be available as a reference for folks after we wrap up today. So and I'll just thank Shannon and Heather for all their help as well. I don't know if anyone has any last minute things to say.
Sounds good. Thanks, everyone. We'll see you next week.
We can leave up the survey information or the QR code if people need that while we wrap up here.
Thanks, everyone. Have a great weekend.
Thank you, Sarah.
Bye.
Thanks, Shannon, for putting the